Hello, everyone. My name is Catherine Heldy, and I'm the Chief Scientific Officer here at Syngap Research Fund. Welcome to today's webinar. It's June 19th, 2025, and I'm really grateful to be able to introduce today's presentation with Professor Pierre van der Hagen. He has written some amazing papers. He's given webinars for a webinar for us before, and we're really excited to have him talk today about linking SYNGAP1 and human-specific genes SRGAP2B/C that control the tempo of synaptic development. So this is a basic science webinar that is really interesting, new things that we have not heard about before. So it's my pleasure to introduce Professor Pierre van der Hagen today. He is a full professor at the VIB Leuven Center for Brain and Disease Research in Belgium. He graduated MD PhD at University Libre de Brussels, Belgium, followed by postdoctoral training at Harvard Medical School here in the US. His research focused on the mechanisms linking development and evolution of the cerebral cortex. His lab pioneered pluripotent stem cell-based models of cortical development, including human mouse brain xenotransplantation. His most recent scientific contributions include the identification of the prominent role of human-specific genes in cortical development and function, and the recent discovery that some of them may act as modifiers of neurodevelopmental diseases, including SYNGAP1 haploinsufficiency. Pierre's work has been recognized by several international awards, including the Remedios Caro Amelia Prize in Developmental Neurobiology, the Roger Spolberg Prize for Neurological Diseases, and the Franqui Prize. A recorded version of this webinar will be available on the SRF website under Webinars, under the News and Events menu. And that link is curesyngap1.org/webinars. By the end of this presentation, you'll have the opportunity to get answers to your questions, and we'd love to hear from you. So please write your questions in the Q&A below in this webinar. For those of you just joining us, welcome. And again, our speaker today is Professor Pierre van der Hagen. His presentation is linking SYNGAP1 and human-specific genes SRGAP2B and C that control the tempo of synaptic development. I apologize for my mispronunciations, and we're going to hear how to actually pronounce things now, since I'm turning it over to Professor Pierre van der Hagen. Thank you.
Thank you so much, Catherine, for the very kind introduction. And don't worry about the pronunciation. Van der Hagen is as you probably notice, is a Dutch name from the Netherlands, and I actually grew up in a French-speaking family, so I don't know how exactly to pronounce this name either. Comes from my ancestors, but I think any way to pronounce it is just fine. All right, okay. So I'm very pleased to be with you again. I think I came 2 years ago or something like that, where we were starting to get interested into the SYNGAP1 gene, and I will remind to everyone how we got interested into it and so on. And I'm really excited to share with you some data that we are pursuing now, that we obtained recently, and that really make us actually pursuing this idea that studying human-specific pathways of brain development may be actually relevant to understand specific neurodevelopmental diseases, such as the one caused by SYNGAP1 haploinsufficiency, and even, who knows, to think actually of therapeutic options, as well as diagnostic tools that could be interesting as well in this context. And this is where I think I would be really interested to understand, to hear your feedback and your thoughts on it as well, even though, of course, as Catherine said, all of this is still very basic science at this point. Okay, so indeed, it's very basic science. It starts with a very basic question that is basically what could be the origin of human brain evolution, and it's well accepted now that a lot of what makes our brain different from the brain of other species comes from differences in development of the brain. And that's what makes the human brain contain more neurons and glial cells, more types of specific neurons, larger neurons, more synapses. But there's one particular aspect of the development of the human brain that more and more people realize could be very critical to understand the development of higher cognitive function in our species, and that is the speed of development. And so what do I mean by that is that neurons of the cortex display a very prolonged timeline, so that to reach maturity, as you can see here, looking at a proxy of their connections, their synapses, their dendritic spines, you see that in the prefrontal cortex, it takes 16 years for these neurons to reach a peak of maturity. And the equivalent in the mouse would take place in about 30 to 60 days. So 2 decades instead of 2 months is basically what we're talking about here in terms of difference of scale of developmental timing. And so as you know, as a question of basic biology and basic developmental biology, there's really this question: how is it that neurons that look very much the same, human cortical neurons, mouse cortical neurons, takes such a difference in timing? And also another question, as always in biology, is not only the how, but also when it comes to evolution, as to the why. So how is it that this protracted, prolonged development was selected? And in fact, what evolutionary biologists have proposed decades ago is that the fact that the cortex is slower to develop than other parts of the body and the brain leads to this so-called neoteny. And by the way, if you don't see my mouse, please let me know. So this neoteny that leads to the, which means basically, literally, retention of juvenile features. It means that the neurons of our cortex remain much younger than the neurons of other parts of the brain and other parts of the body. And that could potentially lead to enhanced plasticity of our brain and lead our brain basically to be better optimized, if you will, to the environment. And this is indeed a characteristic of a human brain development and human development in general, is that it is indeed much more dependent on the environment than in the brain of other species. So that's basically 2 questions we're very interested in the lab. On the one hand, what are the basic mechanisms that explain how is it that human cortical neuron develops so slowly for such a protracted period of time? And what could be the links between cortical development that is so protracted, brain function, and also brain dysfunction? And this is where there is a link with disease. And basically the hypothesis that we make is that if neoteny is really important for what makes the human brain cognitive function and social function so special, well, then maybe disruption of this neoteny in pathological context could happen as well. In other words, what was selected as a positive trait could be somehow disrupted in some diseases. So of course, this is a very simplistic hypothesis, but I hope I will bring you some data today that suggests that indeed it may be the case in some diseases, including in SYNGAP1 haploinsufficiency. So one way by which we study the mechanism of neoteny is this experimental model that we developed over the years, and that is called xenotransplantation. So that literally means we take neurons of one species, like human, and then we transplant them in another species, in the brain of another species, in this case, the mouse. And we can do this thanks to pluripotent stem cell technology that I'm sure all of you have heard before, where we can make cortical neurons out of stem cells. This is actually what we invented more than 15 years ago. But then we can transplant these neurons into the mouse brain and look at their development. And you can see here how these human neurons look like in a mouse brain 1 month after transplantation. Now, when we did these types of experiments with mouse, macaque, or human stem cell-derived neurons, we made this surprising observation that while mouse to mouse, as you can see here, leads to mature neurons in about 1 to 2 months, so that the mouse neurons, when they are transplanted, basically develop as fast as the mouse. When we do the same with transplantation of human neurons, well, you can see that it takes basically months and months for them to develop. And if you take ape neurons, well, they will basically develop along an intermediate timeline. So everything happens as if the neurons develop along the timeline of their species of origin and not of the brain in which they were transplanted. So everything is as if it was intrinsic, internal to the neurons themselves, that there was some kind of clock or several clocks that somehow control the speed of development. And this is a big topic of study in our lab and in many other labs now, try to understand what is this clock. And in fact, a hypothesis that we made is that maybe one of this clock takes place within the synapses of the neurons in relation with synaptic protein. And that's what got us actually to SYNGAP1. So I think many of you know probably more than I do about SYNGAP1 mutations and their link with neurodevelopmental diseases, intellectual disability, epilepsy, autism spectrum disorders. I'm sure you know also about that are caused by haploinsufficiency in SYNGAP1 gene. And you also know that the work of many labs has led to the finding that SYNGAP1 is actually a very abundant protein in synapses. It's involved in their development and function. And work from Gavin Grumbo and colleagues, as well as Rick Huganier and colleagues, had suggested that somehow synapse formation in the mouse is increased and maybe accelerated following mutations of SYNGAP1. And so that lets us to think that maybe there's a link with developmental timing and tempo and the neoteny I was telling you about. And so really what we decided to do is to test whether haploinsufficiency of SYNGAP1 could somehow be linked in a negative way with the synaptic neoteny I was showing you. And this is a work that Ryo Iwata and Ben Vermark, postdoc in the lab, together with Vincent Bonin and colleague here in Leuven, took over. So we first generated mutant stem cells and neurons for SYNGAP1, and you can see here the SYNGAP1 level of protein that become half of normal, so mimicking the haploinsufficiency. And then we derived cortical cells from these, and we transplanted these cells, and we looked at them in a variety of ways. And I'm going to show you a number of experiments now, first focusing on the neurons themselves, their morphology, their physiology, and then, and that's more recent data that I think are quite interesting maybe to understand the pathophysiology of SYNGAP1 haploinsufficiency when we look at the function of these neurons within cortical circuits in the mouse, but using human neurons. So the first type of data I'm going to show you is basically looking at the morphology of development of these SYNGAP1 haploinsufficient neurons. And what we found basically is that if we follow the development of synapses in control neurons, you can see in 4 months and 7 months of development that it goes quite slowly. You still are far from reaching a plateau with control neurons. But if you look at mutant neurons, you can see that they have many more synapses, many more dendritic spines already. And this is what's quantified here. So the speed basically of development of synapses is faster in the SYNGAP1 heterozygote neurons, so suggesting that SYNGAP1 haploinsufficiency somehow disrupt the slow development of neurons. And we've looked at this in many other ways, and I'm showing you here another way where we could do physiology measurements of the synaptic activity of the transplanted human neurons, and we could see that basically their synaptic activity is also accelerated. So we can see basically formation of synapses here from frequency of synaptic currents that is increased, especially at early time points, so suggesting acceleration of synaptic formation, and also other parameters here like the AMPA and MDA ratio that is again accelerated, that develops in an accelerated fashion in time. So if you put all this together, it suggests that the synapse development speed is indeed higher and that it's accelerated in SYNGAP1 heterozygote human neurons versus control neurons. So suggesting basically that SYNGAP1 acts as a sort of molecular brake to slow down the clock of development, so that when you remove it, well, the clock goes faster. Now, a big question in developmental neurobiology is actually, okay, timing of maturation of synapses is accelerated, but does that have actually a consequence at the level of the cortical circuits? Because after all, it's at the level of circuits that important things take place, including at the level, of course, of the disease. So we asked what could be the consequences of the accelerated timing of synaptic development at the neuronal circuit level, but still looking at transplanted human cortical neurons in the mouse. And so for that, we developed another tool, in particular, thanks to Vincent Bonin, and that I'm explaining to you now. So in this experiment, we basically transplant human neurons in which we inserted a gene called GCAM that enables to follow the function of these neurons. And we then do multiphoton microscopy. This is a super microscopy that we can do in living mice, and where we can look with this genetic reporter at the activity of human neurons in awake mouse. And I'm going to show you here a movie where you can see 1, 2, 3, 4, 5, 6 human neurons in a mouse cortex circuit. And the mouse is imaged with our microscope, and at the same time, it's looking at the stimuli that you can see here that are stimulating its visual cortex. And these neurons here are in the visual cortex. And what I hope you can see is that each of these neurons is actually sensitive to specific stimuli. Sorry, I'm going too fast here. So each of these neurons is specific to these specific stimuli. And that's a process that is actually a characteristic of our visual cortex, which is called tuning, meaning that each neuron in our visual cortex is sensitive to specific stimuli. And this is basically how we build visual images in our brain. And so quite surprisingly, quite remarkably, I would say, to us, the human neurons transplanted in the mouse brain are able to show this very substantial level of functionality. So we can mimic in a way in the mouse brain how human neurons would function in the context of visual sensation, perception, and cortical circuits. So we applied this system to control neurons and SYNGAP1 haploinsufficient neuron. And what we discovered is shown to you here. Control neurons early on, 4 months after transplantation, show very little stimulation. So the stimulation is shown in red, and you can see very few of these neurons that are sensitive to anything. They also show very low level of tuning, meaning that they're not sensitive to anything specific. If you wait long enough, 3 months later, you can see that now they can become tuned to specific stimuli. Now, if we look at heterozygote neuron, well, you can see, if you look at heterozygote neuron, they are precociously stimulated by the stimuli. So they show precocious visual responsiveness. And this is a quite robust phenotype that suggests to us that when SYNGAP1 is lost on one copy of their genome, well, these neurons develop basically sensory responsiveness too early for what would be required. So could that have additional detrimental effect? So to test that, we looked at another classical way to look at the sensory function, but in this case, looking at sensory plasticity. And I'm depicting you here classical experiments of the Nobel Prize winner Hubel and Wiesel, who discovered many, many years ago, in that case using cat visual cortex, that there are columns in our brain, this is also true in human, that are sensitive to either the left eye or the red eye, and these are the so-called ocular dominance columns that enable the vision the vision of in-depth, the 3D vision, basically in our brain. So we tested basically what's going on with this in our mice. I'm sorry, I'm going too fast here. What they show, what Hubel and Wiesel and colleagues showed, is that, so these are columns that are sensitive to one eye or the other. And what they showed is that if you close one of the two eyes, the part of the cortex that was normally sensitive to that eye lose responsiveness to that eye and gains responsiveness to the eye that remained open. And that is what is called monocular deprivation plasticity. And this happens in a very strict window of time that's called critical period. And that in human is basically taking place during the first postnatal years, and in mice during the first postnatal weeks. And so we asked, what's happening if we put human neurons in the mouse now? Are they going to show this plasticity? And what's happening to human neurons that are deficient for SYNGAP1? Are they going to lose this plasticity? And the answer is yes to each. So now here are the data. Again, we take our mice with this genetic tracer of activity. We will measure their activity before we close one eye. Then we close one eye for a few days, and we ask what's happening to them after having closed these eyes. So we measure the activity of the same neurons over time. And here is an example of a neuron that was sensitive to the left eye. Then we close the eye, and after eye closure and opening it again, you can see that it lost responsiveness to this eye. So this is what the plasticity is about. And it's a very classical model to study plasticity, sensory plasticity in that case of the cortex. And what we found is that at the level of all neurons that are transplanted, when we transplant control neurons, you can see that the majority of them show this shift of plasticity. But if we do the same experiment with SYNGAP1 haploinsufficient neurons, well, you see that they lose this plasticity. So this suggests to us that the SYNGAP1 haploinsufficient neurons that are developing precautiously, that are developing too fast, well, one of the things that happens then is that their synaptic connectivity gets basically matured so fast that they lose the ability to respond to monocular deprivation and to respond to the environment. And we think that perhaps this disrupted plasticity may reflect what may happening now in the brain of SYNGAP1 haploinsufficient patients, even though, of course, this is something that needs to be studied in much more detail. But that suggests to us that indeed maybe the disrupted synaptic neoteny and the disrupted neoteny of neurons at the level of circuits could be something that would be one of the pathogenic mechanisms very early on that would take place in SYNGAP1 haploinsufficiency. Now I'm going to switch gear, and if you have any question at this point, please feel free to ask me. But if there's not any present question, I can now switch to a second part of the talk, when I want to tell you now where, using this model of SYNGAP1 haploinsufficiency, we try to understand better at the molecular level, the genetic level, what is the link between SYNGAP1 and human-specific neoteny, human-specific prolonged developmental timing. And basically, the way we try to tackle this is, yes, as I just said, we try to understand what could be the molecular mechanism underlying these processes. And to do that, I need to go back a little bit to basic science, to very basic science, and tell you that something that we and others discovered several years ago now is that one of the reasons why cortical development is different in human from development of the cortex in other species is because our genome, our human genome, does not have all the same genes than the genome of other species. We share most of the genes with other species, but there are exceptions. And these are typically genes found only in the genome in the human because early in hominids, so great ape evolution, in our ancestors' evolution, there was duplication of these genes. And that somehow led to new genes that have new functions in the human and not in other species. There are many of them. You see all their list here. You don't need to remember all of them, of course. But there's one family in particular that we got very interested, and that is going to be very important now to focus on, because this is the gene family that we discovered is actually acting together with SYNGAP1 to control neoteny. So what is this family? Well, it's a family of human-specific genes that was discovered actually by our colleague Frank Polleux in Columbia many years ago now. And what they discovered at the time is that it's a gene family. There is one member of the gene that is present in all mammals. It's called SRGAP2 or SRGAP2A. And in human, we also have the same gene that we inherited from our ancestors, but our very recent ancestor also duplicated that gene so that we have several human-specific copies of SRGAP2. And what Frank discovered is that if we overexpress SRGAP2B and C, the human-specific gene, in the mouse, we delay the maturation of the synapses in that mouse. So we induce, if you will, something that looks like neoteny. The synapses develop slower in the mouse because of these human-specific genes. So together with Frank, we thought, what if these genes are actually involved in human synaptic neoteny? And to test that, we decided to look at these genes in human cortical neurons. And that's the work in particular of Baptiste Libet-Philippot, another very talented postdoc in the lab, together with Franck Polleux and Cécile Charrier, a former postdoc of Frank's lab who is now in Paris. So what we did basically is the same experiment I showed you with SYNGAP1, but now instead of removing a copy of SYNGAP1, we downregulated, we removed basically the expression of these human-specific SRGAP2 genes. And we asked, how do synapses develop following transplantation in the mouse of these human neurons? And you can see here again the very prolonged development of the human neuron. You see how much time it takes for the synapses to appear. And here is now the very spectacular result we obtained when we remove SRGAP2C. You see that they basically develop in almost 6 to 12 months earlier than their control counterpart. And on the other hand, what we discovered is that we remove SRGAP2A. So that's the gene that's common between mouse and human. We see the opposite effect. You see they go much slower. So you see here a summary of what we found. We remove the human-specific gene, everything goes faster. We remove the gene that is also present in the mouse, everything goes slower. And in fact, at first we were surprised, but in fact this goes in line with other data that were obtained by Franck Polleux and other labs, that SRGAP2C genes, they actually act by inhibiting SRGAP2A. So in fact, what happened in human evolution is that somehow new genes appeared that act as brakes, if you will, of the SRGAP2 genes, and these are the SRGAP2C genes. I understand it's a lot of information, but it basically shows you that on the one hand, there is SRGAP2A gene that promotes synapse formation, so that when you remove it, it goes slower. And then in human evolution, there is this brake that came out, and when you remove the brake, it goes fast. Now I will show you more data that tackle this and that back up this model. That just to show you that the same happens at the synaptic level, but that's kind of a detail I would say. That shows you a summary of the data again. Control neurons are slow. We remove the mouse and human gene, it goes slower. We remove the human-specific genes, it goes faster. And it's the same as when we remove one copy of SYNGAP1. So could it be that SRGAP2 and SYNGAP1 are linked now? And that's what I'm going to show you now. And it's some molecular data. I'm not going to show you all the data here because I realize that this is really heavy-duty molecular biology. But basically what we did is to extract the synapses of neurons that have normal level of SYNGAP1 or abnormal level of SYNGAP1, that have normal level of SRGAP2A or abnormal level of SRGAP2A, that have normal level of SRGAP2BC or abnormal levels. And to make a long story short, I'm going to show you immediately the conclusion that we come with, and I will show you some of the data after. But the conclusion that we come with is that SRGAP2A is regulated by the human-specific SRGAP2BC. But SRGAP2A itself controls the level of a SYNGAP1. And this is how we think synaptic neoteny is controlled. Now, why do we come to that conclusion? Well, because when we look at the synapses of the human neurons, if you look at the level of SRGAP2A first, you see that they are, here is the normal level. You see that they are removed when we manipulate the level of SRGAP2A with genetics, so that's expected. But you see that they are increased when we remove SRGAP2C. So this is this negative arrow here. And it shows you how SRGAP2BC sort of remove the brake that SRGAP2A constitutes. Now, look at the synaptic level of SYNGAP1, and you can see that the level of SYNGAP1, here are normal levels, they go up when we remove SRGAP2A. And this is what's shown you here, this negative arrow here. And as a result, they go completely down when we remove SRGAP2C, because what SRGAP2C is doing, as you can see here, is to remove SRGAP2A. So I know it's a bit complicated to follow, but it means basically negative regulation of a negative regulation becomes a positive regulation. So those human-specific genes are required in human neuron to have normal levels of SYNGAP1. And that, I think, is a very important result also in the context of SYNGAP1 haploinsufficiency. Because indeed, the other thing that we discovered that's shown to you here is that in SYNGAP1 haploinsufficient neuron, where, as expected, the level of SYNGAP1 are very low, if we remove SRGAP2A at the same time, they go back to almost normal levels. In other words, SRGAP2A, because it's a negative regulator of SYNGAP1, can be a target to increase the level of SYNGAP1. And I'm going to show you that this is not just about biochemistry, it's also about synaptic function. So we have done the same experiment, but now looking at synaptic function. And to make a long story short, what we found is that, again, if you look at synaptic development in SYNGAP1 mutant, you see how it's accelerated. And you can see that if we remove some levels of SRGAP2A in the SYNGAP1 heterozygotes, well, the acceleration goes down. So the speed of development is decreased. And it's even more true if we look at synaptic activity that is abnormally going up when SYNGAP1 is haploinsufficient. But when SYNGAP1 is haploinsufficient, but we also downregulate the level of SRGAP2A, you see that the levels of synaptic activity go back to normal. So I know it's a lot of data, but I think we can put them all together with this model that I show you here now, and which is a model that in biology people like a lot, to use the model of a balance. And indeed, I think it makes a lot of sense that normal life is about balance and pathology is often about an imbalance. And we think that really what goes on here is that there is a normal balance of SYNGAP1 and SRGAP2 that controls the timing, the tempo of synaptic development. And this balance is different in human because of the SRGAP2B gene that tip the balance by decreasing the level of SRGAP2A to a tempo that is slower. So that's, we think, one of the reasons why synaptic development is slower in human than non-human. But then in SYNGAP1 haploinsufficient neuron, well, they go faster because despite the fact that as humans we have SRGAP2B genes, if one copy of SYNGAP1 is removed in SYNGAP1 haploinsufficient mutant patients, SRGAP2A levels goes up. And the balance then goes to faster development. And that would explain also why, if this would all explain why in SYNGAP1 mutant neuron everything goes faster that I showed you. But if we also decrease the level of SRGAP2A, well, at least the speed of synaptic development can be brought back to what seems like normal levels. So if I summarize everything that I told you, I showed you that on the one hand, we found that SYNGAP1 haploinsufficient neuron show a disruption of neoteny, and maybe the slow development that is characteristic of the human species, maybe something that we can try to understand better and work on in the context of neurodevelopmental diseases like SYNGAP1 haploinsufficiency. And indeed, human-specific genes like SRGAP2B can modify the course of SYNGAP1 haploinsufficiency in our experimental model. And of course, this is a question mark, but it could be proposed at least that SRGAP2B genes could constitute potential targets, either diagnostically or therapeutically, in liquid SYNGAP1 haploinsufficiency. And of course, it's still a long way before we get there, but we are actually now launching a project together with the lab of Franck, where we're going to test this directly. So we're going to look not only at synapse development, but also at the function of the human neuron transplanted using visual stimulation and visual plasticity, as I showed you before, as well as in mouse model of SYNGAP1, to see whether changing of the level of SRGAP2 can somehow modify the abnormalities that are found at the circuit level in those experimental models. And that would be the next step of our research, bringing more, let's say, towards what I would call a preclinical approach of the data that I showed you that are still, of course, still very basic. And so with that, I want to thank again all the people in the lab who are involved in this work, in particular Baptiste, Ben, and Rio, as well as I mentioned Franck and Vincent, as well as all the amazing grant funders who have helped us to build up this work, and that we hope to pursue in the future, more trying to link the basic mechanism of human brain development with the biology of SYNGAP1. And with that, I will stop and probably stop sharing, and thank you a lot for your attention.
Wow, just wow. That is absolutely amazing.
Thank you.
I will definitely be watching this again. Let's see, before you stop sharing, do you want to, let me just read one question here. I have a bunch of questions, but I'm going to be a good host and I'm going to ask the question typed into our Q&A. So this is Balaji Shriram. Oh, let's see, actually. The first one was, thank you. This was a great talk. The spine counting exercise, so this is in the first half. The spine counting exercise should indicate that the effects are primarily in excitatory synapses. Were you able to test the effects on inhibitory synapses separately?
Yes,
and what could cause this differential effect?
Yes, so that's an excellent question. So we have not been able to count inhibitory synapses because, as you may know, this is even more difficult than excitatory synapses because they are not spiny. But we could look at inhibitory synaptic currents. So we could look at the activity of inhibitory synapses using frequency of iPSC currents. And there we could see also an early acceleration of their development. So it's interesting because it suggests that there may be some kind of attempt of the SYNGAP1 haploinsufficient neurons, if you will, to adapt to the hyperexcitation by also accelerating inhibitory synapse formation. On the other hand, we know that the SRGAP2 genes are not only in excitatory synapses, but also in inhibitory synapses. That's a work that was performed by Franck Polleux and colleagues in the mouse. So that may suggest that some of the imbalance of excitation and inhibition that may take place in SYNGAP1 haploinsufficiency, including in patients, and that may cause epilepsy, for instance, as you know, may be linked to the fact that SYNGAP1 and SRGAP2 may have a differential effect on inhibitory versus excitatory synapses. And that's something else, actually, that we would be very interested to study in the future at the circuit level using mouse models and perhaps human models when we have developed more tools to look in more detail at their inhibitory innervation. But thank you. This is indeed a great question to look at.
Thank you. And then Balaji has a second question. Also want to understand the xenograft model. Do the grafted iPSCs integrate only in cortical areas? If not, what can they say about effects elsewhere?
Yes. So one quick thing, just to be clear, we do not transplant iPSCs. We transplant the neurons. So the iPSCs are first differentiated into neurons, and then and only then they are transplanted. In a way, we are trying to do something similar to what people these days are trying to do for therapeutic transplantation in Parkinson's disease, if you will. And it's very important because I'm insisting on this because if we transplanted stem cells, all we would get would be tumors, actually. But what we get are those differentiated neurons because we transplant young neurons in the first place. Now, can they transplant elsewhere? Yes, they can, but they do not migrate as well as they do in the cortex, because the trick that we use is to transplant them in the ventricule just below the cortex at the time of development, at birth, where there is still what is called radial glia that enables the neuron to migrate and embed into the cortical gray matter, and thereby later on into the cortical circuits in a physiological way. While if they are transplanted elsewhere in the brain, which happens by accident, they can survive, they can differentiate. We could even show that in that case, they can make synapses. They can also send axons and make synapses with the rest of the brain, but they will not get the type of physiological responses that I showed, like the visual responses, because they don't get the right input. So that's why we mostly focus on the neurons that are well integrated and transplanted in the cortex. So because the other neurons, even though they are able to differentiate to some extent in a normal way, they don't develop as let's say, as far as they can in the cortex, where they also get the right input, the right environment, and where they mimic much better, we think, what's going on, let's say, in a real brain.
Amazing. Thank you. I can tell you guys are incredibly specialized, incredibly observant. You know, you're really talking about quite specialized observations. Okay, well, I have a bunch of questions, and I just for the sake of the slides, I just am going to ask like not the most important one first, just so we can put the slides up and then take them down. So I was really interested in your Western blot, where you at the very beginning of Syngap, and you showed 3 bands.
Yeah, we can go back to the very first one. Yes.
Yeah, the very first one had 3 bands, and then this one is more of like a 2 band. So yeah, so here where your arrow's pointing to Syngap1 and the plus minus.
Yes, absolutely. Yeah.
Do you have a set? Is the top one the A alpha 1 form and the second one the B form? Like, do you know anything about that?
That's a great point. Yes. So this is a question that we've been asking ourselves, and that we discussed actually also with Rick Hugen here, is, you know, that this would be a nice model to try to distinguish in human developing neurons, especially in the context of circuits, you know, which isoform is used preferentially, because I think that's very important clinically as well, as I'm sure you're aware of. But unfortunately, we didn't have the tool at this point to know whether this is what we're looking at here, so a different isoform, or whether it could also correspond alternatively to different phosphorylated form, because as you know, SRGAP1 is phosphorylated, and that could also lead to these differences. So we often see these 3 bands. We see them sometimes, sometimes not. We're not, you know, hardcore biochemist of the synapse, so it could be, the variability could come from techniques, but it could also come from biology, right? That the state of the neuron might be critical. What I can tell you, what could be about, maybe it's not related to the question that you asked, but maybe it is in a way about, let's say, heterogeneity of Syngap1. What I can tell you, and that's data that we have not published yet, and that also we mentioned to Rick, and I think it's really, really intriguing, is that when we do immunostaining of the spines with Syngap1, the transplanted neurons, we don't see the same level of Syngap1 in every synapse. There's a heterogeneity of the synapse, and it's correlated inversely with the heterogeneity of SRGAP2. So what I showed you, this tug of war between those 2 synaptic proteins, probably takes place at the level of individual synapses, and may have to do, for instance, with the level of plasticity of individual synapses. So it's one more level of complexity, if you will, but I think it's something really, again, to bear in mind to understand the full meaning, let's say, of what Syngap1 does dynamically in a circuit, and also what happens dynamically in a circuit when half of the protein is gone.
Right. So I mean, I think with the haploinsufficiency, it's always interesting. There's, you know, it's a pretty simplistic idea. You have half the amount, right? But then you start looking at it and you're like, well, this is actually quite complex. So I'm always interested in the fact that we have half the protein, but then there are more synapses that mature too fast and are too big, which sounds like there's too much protein, right? Like that, like just in a naive sense, that sounds like too much. But over and over, we see the Western blots and the Northerns that show that, yeah, there's really half the amount here. So, and then that's one sort of like simplistic idea that has like a little bit of what's happening here. Then you layer on top of that your whole idea where you just showed this beautiful data about a gene duplication, where then the duplicated genes are actually antagonizing its parent gene, right? So these sequences are going to be very related, right? Very related sequences, but now they're kind of having this opposite effect on each other, antagonistic effect. And so you can think of that with a gene duplication. You can also think of that with isoforms, right? Like you can have isoforms of one gene antagonizing each other. So I'm just saying this is all I love getting into the weeds here, and I don't have a simple story yet in my mind of how to explain all this, but I feel that you're really getting somewhere with showing us this.
Yeah, thank you for bringing this up. This is something actually that I think is super important and super interesting. And in fact, we had in fact, if you want the whole truth, it's kind of sad, but anyway, we're trying to we have a project actually where we would like to not test first isoforms, but test at least different domains of the SYNGAP1 protein, because that's the other level of complexity, right? Is that SYNGAP1 is not just one tool, it's actually a protein that does many things at the synaptic level. And these things may not necessarily all be synergistic. Some of them may be antagonistic, some may be in parallel. And so we have developed a system where we can, in principle, see whether we can rescue the defects with specific parts of SYNGAP1 protein in human neurons, and then ask which domain is important. That could be relevant also to the isoforms, because as I'm sure you know, the isoforms have different domains as well. And so that would be another way to get into that. The reason why I'm saying that it's a bit sad is that we were hoping to get funding for this from the NIH. And as you know, all funding for foreign investigators has been completely disrupted. So we'll have to find funds elsewhere for that. But I don't want to insist on this, because I think the situation of US-based investigators is much more serious. And that's the one I think we should complain about and focus on. But we had very good feedback from colleagues who work on SYNGAP1 and who told us that indeed, as you say, this is actually a very important step maybe to look at afterwards, is to see individual isoforms and individual domains of SYNGAP1 to see actually what really matters in the observations that we make.
Yeah, thank you. And thank you for bringing up the funding issue. And it's amazing. We all need to hear what the repercussions are.
Bring it up because I think
we need to hear. Yeah.
It's really terrible right now in the US, but anyway, it's another problem, I guess.
Yeah, absolutely. So I don't see any more answers in the Q&A. I mean, any more questions in the Q&A or the chat. So I hope people don't mind. I'm just going to keep talking. Is there a way we can, are you sharing, or oh, there we go. Okay,
I stopped sharing. Is that okay?
Perfect. Yes. No, that's perfect. It just took a moment for my screen to catch up.
Sorry. Yes.
Okay. So, I have a couple kind of broader questions that I hope if you don't want to entertain them, please say next. So this, one thing that's really interesting about humans is this really extended neoteny time, right? And I just wonder if, I wonder if we can talk about that in terms of not just when the neoteny. So as a developmental biologist, I love hearing about this time, right? This is like early. Usually developmental biologists talk about early development. But I'm also wondering about late development, like senescence. And so I'm wondering about things like Alzheimer's or other diseases that might come into play here. So what can you comment on that a little?
Yes, I can try. I mean, so I'm not an expert of Alzheimer at all. Um, so I think there's obviously always this question, is our development prolonged just because we have a longer lifespan, and which also makes us more sensitive to get demented? I mean, that's what
Mhm.
so I don't, and you cannot really say no to either of these. But what I can say is that the scale of timing of synaptic development is really out of range compared with the scale of increased lifespan. If you understand what I mean, is that, okay, yes, we have an increased lifespan compared with other species, but we have a much increased span of neoteny compared with other species. So this is really why we
Yeah.
can talk about neoteny and not just that, everything is prolonged. Now, to go back to your second question, so people have not looked, to my knowledge, at these genes in the context of neoteny and in the context of a neurodegeneration. But I do know, and that I think is an exciting venue, and I don't think I'm going to study that, but I think other people would be very well placed to do it. There is evidence now from the lab of Frank Polleux that SRGAP2 genes, not SYNGAP1, but SRGAP2 genes, are also expressed in microglia. And as you may know, and it plays a role there in the control of the function of microglia. And as you may know, microglia function has been now, I think, not only proposed, but shown to a large extent to be quite critical in some of the early phases of dementia, and as a part of the neuroinflammatory response that takes place there. So it is tempting to speculate that the same genes that are involved in synaptic neoteny may have something to do with microglia biology as well, also in the context of pathology. And that so that somehow they are not reused in the context of neurons, but maybe reused in the context of microglia when there is neuronal loss. But that, for now, there is zero data for this.
Yeah.
But I
Yeah.
want to convince my colleagues who study microglia and SRGAP2 to look at
Yeah.
this in the context of AD.
And I mean, my mind goes to pruning. Is that the obvious thing to think of?
Yes,
Yeah.
yes,
Okay. Okay.
exactly. Pruning
Okay.
could be that the pruning that microglia are supposed to do in development, and that may have to do with neoteny as well. One quick word I can say about this. We would love, of course, to be able to look at pruning in our model. The problem is that pruning in human cells is probably taking place even later.
Mm.
And so there is currently no good cellular model of synaptic pruning using human cortical neuron, because we would need basically to have human neurons that would correspond to the second, the third, the fourth postnatal year, while we at best probably model here, I would say, the first 12 months after birth.
Yeah. So you'll need a different model, which maybe would be maybe slices from surgeries. Is that the idea?
Exactly. And
Yeah.
that's actually something that we are
Yeah.
exploring to study as well. Yes,
Yeah. I
exactly.
think those can last up to about 4 weeks, maybe. So I don't know if
Absolutely.
is that enough time, do you think, to do these types of
Could be.
experiments?
We have been talking so we have actually ongoing in the lab other projects on human cortical neuron function. And together with Frank, we thought maybe when we have some good cases, we could try to knock down SRGAP2 or
Mhm.
SYNGAP1 in an acute fashion, because that, to my knowledge, would be the first way to look at synaptic function in a way that is as close as possible to what's going on in the human brain. But as you
Yeah.
know, these are really hard to get, so it's not so easy
Right.
to
Right.
set
Right.
up these experiments, of course.
Yeah. Well, it sounds like hard experiments don't scare you. So,
Very
um,
important to do in the long run, yes.
yeah. So I should have looked this up before the webinar, but is SRGAP2A or any of the SRGAP2 genes, do any of them have a monogenic human disorder associated with them?
No, they don't. They don't, which again, I think is interesting if you think of it as a potential target, right?
Mm-hmm.
So it's fixed in the population. So it's very important, of course, to have a SRGAP2A. You don't have individuals around with only one copy or
Mm-hmm.
zero copy. So it's a very important gene, very conserved among mammals, but there is no disease associated with them at this point. There was at some point data from Evan Eichler suggesting that loss of SRGAP2B I'm sorry, SRGAP2C, so one of the human-specific gene could be associated with intellectual disability, but it's a single case of many years ago, and since it was not confirmed, a single case in human genetics is just not enough, right? So we don't know.
Well, at first it's not enough.
Sorry?
At first, it's not enough. Later, we'll go back and say like, oh yeah, that was the first one.
Exactly.
Yeah.
So
Yeah.
we will see. We will see. Human-specific genes are really tough guys to study in human genetics.
Mhm.
People like Evan Eichler are doing big efforts to develop new technology to sequence the genome in a way that is more appropriate for that in cohorts of neurodevelopmental diseases. So we may get more data on this, but for now there is no data.
Yeah. So are they in the new? Well, anyway, I could go on forever about the genetics. I will stop there and just say I look forward to lots of population genetics on these genes, right? To see what's the variation in well population, and are there small changes that actually are disease?
Well, we know they are fixed, huh? So that we know. So those human-specific genes that we study, we know at least their copy number is fixed. So that would suggest they're important. But what's happening if you mutate them is the thing that is unknown.
Yeah. Okay. We do have another question, which I should read to you. This is Nick Murata. Thank you for such an interesting talk. I was wondering if you had any thought of utilizing cortical organoids as a parallel model, or if you have any thoughts on this model to potentially generate neurons that you then transplant for your studies.
Yeah, these two great questions. So, as you know, well, because of all I explained to you, the big problem of modeling any type of neuronal development is time. Is that if you want the neurons to be studied at a level of synaptic function, synaptic plasticity, and so on, you need to find a way to get to the level of maturation that I showed you. In our hand, the only way by which we are able to do that is using xenotransplantation. For instance, to look at dendritic spine formation, synaptic function, and plasticity at the level that I showed you, we have not been able to find in organoids. That said, there are other groups that are trying very hard to get to that level. But if you think, for instance, of the work of Sergiu Pașca and colleagues at Stanford, he showed quite compelling data that spine formation, synapse formation from the same cells, from the same organoid, if they are transplanted, can become much more mature. So indeed, there is probably in vivo context that is very important, and which is the reason why we think xenotransplantation is important. On the other hand, we are exploring whether we could combine xenotransplantation to start with, for instance, to look at the generation of more specific types of cortical neurons that are generated more efficiently using organoids than in 2D models that we use, and then transplant these neurons in the cortex. So that could be used, and we've discussed this with Sergiu, and I must say that there are pros and cons of the two ways to think of it. This is becoming a bit technical, but you can either transplant the organoid as a whole, but this can only be done in rat because the organoid is too big to put in mouse. So that's a technical hurdle for us because we do a lot in the mouse that cannot be done so easily in the rat. And on the other hand, the other thing, at least that's my take on it, is that because we dissociate the cells first, but we could do that from organoid. We've done that. We can dissociate the cells from organoid and then transplant them. As I explained earlier, they are embedded in the cortical circuit of the mouse. And I think that makes them an easier path to function because their input will be much more specific than if they are in an organoid separated from the cortex. It sounds a bit trivial, but I think this is part of the thing that remains, let's say, a difference between a model where you would transplant the whole organoid or where you would put the cells within the cortex itself as single neurons, if you will. And we have written a review about this actually in Cell a few years ago, and I think it really depends on the question that you have. If, for instance, you want to do transcriptomic analysis or epigenomics analysis, I think you probably find an even better by transplanting whole organoids because you're going to have many more cells. So it's going to be much easier to do hardcore molecular biology like single-cell transcriptomic. If, on the other hand, what you want to do is fine-grained physiology or plasticity, like what I showed you with the GCamp imaging and so on, I tend to think that you're better dissociate first the cells and put them in the cortex, because they will probably show more tuned responses that are closer to the real physiology than they would be in organoids. So, and to me, this is really my strong belief. In our lab, we use all models possible, and then depending on the exact question, we apply a particular model. And I think for human modeling, this is going to be like this for many years to come. There will not be a single canonical model that everybody uses, you know, like the C57 black 6 mice that everybody uses. There's not going to be such thing with stem cell models. We're going to have to use many models in parallel, or I would say orthogonally, and each of them will bring us a piece of the ground truth, hopefully. That was a very long answer. I'm sorry.
Are there, this is a super naive question. Are there any chimeric, can you make chimeric mice with iPSC, with undifferentiated iPSCs?
I mean chimeric from very early embryos. That's what you're talking about.
Yeah.
Yeah. So obviously, first of all, ethically, we would need very different guidelines. We need
right. I mean, are there even guidelines on that? Maybe the guidelines don't do that.
Oh yeah, they're absolutely very strict guidelines. So with the stem cells that we use, we are completely forbidden to do it. I see.
Okay.
That said, we can talk about it. I mean, I think guidelines, of course, depend on what could be gained out of it, right?
Well, yeah, because my first thought was maybe you could put more of an organoid into a mouse in utero, like earlier brain before it's born. But I thought that first, and then I thought, well, I'll go back farther, and then I thought that's probably bad.
I can do a quick comment on this. Two quick comments. First of all, if we tried very early on, even if we were authorized to do it, it would not work, and because in fact the mouse and the human stem cells have such a different speed of development that no matter what you do, the mouse cells take over. Yeah.
Okay. Yeah, makes sense.
Yeah.
Yeah, I told you that was very naive. I'm sorry too.
No, it's not naive. I think it's a very good question. We thought about it, but it remains difficult at this point. What we could try to do, and that's something, but we need to convince somebody daring enough in the lab to do it, because it's a very risky experiment and a little bit provocative, even though I don't think personally that it is ethically sensitive. Personally, I don't think it is, because what makes the human brain is the connectivity and not single neurons. But that's my belief. I can also imagine other people have another opinion. But so you could imagine to use mouse models in which there are mutations that make cortical progenitors of the mouse develop slower or less efficiently, and then transplant these mice. So people have done that with rat embryonic stem cell derived pancreatic cells. Rat into a mouse mutant that don't make a pancreas. And they could make a mouse where the whole pancreas is the rat. And it has the size of a rat pancreas, interestingly. And it's in a mouse. Now, of course, we don't want to make a mouse with a human brain. That doesn't make any sense. But we could imagine, let's say, instead of having a few thousand neurons like what we do, to have maybe a visual cortex area that has 30,000 neurons, so 10 times more. That would still be a minority. It would still be a mouse cortex. But we could imagine to do many more interesting experiments in normal and disease context that I think would be a game changer in biomedical sciences. So that is, I think, an effort that would be worth doing. But again, it's a lot of technology involved. And then at some point, of course, we will have to make a threshold when it comes to ethics. But my take on it is that we are really far away technologically of any, concerning ethical questions. It's always important to have it in mind, but I would say that not right now and not before at least a decade, in my opinion.
Yeah. I mean, it just brings me back to liking the surgical tissue.
Yes,
I think surgical tissue is the most reasonable model, most similar model. Okay, so I'm going to ask you one last question. This probably pulls back to my actual job as the CSO. I'm gonna get away from all the really fascinating things about SRGAP2, A, B, and C, and just say, is there a way that any of those would be a biomarker for SYNGAP1 related disorder? So either at the protein or RNA level, is like looking at those, is that likely to change with the SYNGAP1 haploinsufficiency either over time or with treatment? And where is it, do you think it would be in CSF, or is that, do you think that's not really an accessible molecular signature?
So, I mean, we haven't looked in CSF, so I cannot answer your question in a really informed way. What I can tell you is that in all the experimental models that we've looked at now, in the human neurons as well as in the mouse, actually, SRGAP2A is indeed a quite reliable marker of what's going on with SYNGAP1, going up when it goes down, going down when it goes up, at the single synapse level, at the whole neuron level, at the population of the neuron level, at the whole synaptic level. So in that sense, this is where I'm saying it's an interesting target to think of in terms of diagnostics for sure, and maybe therapeutics, because in that sense, at least, you could imagine also that it's expressed in other cells, maybe other than brain cells, and where it could tell you something earlier than when symptoms occur, for instance, which I think is a big challenge for many neurodevelopmental diseases, right? And it could that you know early enough that there is SYNGAP1 haploinsufficiency, and also to test, of course, potential treatments. But I don't know enough, let's say, about the biomedical context of CSF and biomarkers to give you a straight answer. But to be honest with you, you are not the only one to ask me this question. And in fact, you know, a year and a half ago, when I was talking about this, I was thinking of it essentially, let's say, as a basic science discovery that would be interesting to put in context of understanding the disease, and that's it. But I got quite some feedback of people who are more toward the translational, and who have been asking me questions like the one you ask, and even like, would it make sense to try to knock down SRGAP2 and so on?
Right.
And at first I was very, you know, skeptical as a bike scientist should be, I think, saying, well, no, I don't think so, and so on. No, I'm at least more open to the idea, and I think especially with the data we have at the whole circuit level, and the fact that the biochemistry is very robust, I would say at least it could be explored. And I think really here it's more about people who are more competent than me, let's say, to look at this in the right way. And of course, I would be very open to help in any way I can to try to get to that, let's say, more translational aspect, just to test things, right? I think that would be interesting. Yeah.
Yeah, yeah. Well, SYNGAP1 is, of course, my first and primary love, but there are other things that have synaptic plasticity reduction, and so it's really interesting to think of it as maybe a more general.
Actually, yeah. So I can comment on that actually very quickly because in fact I forgot to mention it in my talk, and I think it's quite remarkable. I mentioned to you that we did in collaboration with Franck Polleux and a former postdoc of his, Cécile Charrier in Paris. In fact, 2 years ago, so she basically collaborated with us in the sense that she gave us the trick to knock down SRGAP2C and SRGAP2A independently with the right knockdown. And then, you know, she was working in Paris, we were working in Leuven and with Franck and so on. And at some point, we discussed first by Zoom and then in real, and we realized that she also found a link between SRGAP2A and SYNGAP1. Coming from a completely different perspective, it was proteomics of synapses and so on. To make a long story short, we basically published back to back the same observations, and we helped her actually to look in human neurons at the same thing. But what she also found that we had not found, that's really her thing on top of it, and that goes in line with what you say, is that she found another protein, synaptic protein, in relation with SYNGAP1 and SRGAP2 that is called CTNND2. And I didn't talk about it with you because otherwise I think it would have become overwhelming. So delta catenin 2. So that gene also seems to play a role so it's not a tug of war, it's a trio, okay? And with D'Artagnan being the fourth musketeer from human specific being SRGAP2C. Okay, so it's full protein. Delta-catenin 2, when mutated in human, also leads to intellectual disability. It's the syndrome of Prix-du-Chat that can be associated as well with ASD and epilepsy. So it does not, we looked at that because at first, the first thing I thought is asked to clinicians, you know, would this be the same syndrome that people have overlooked? No, it's not. So it's a different disease, but it has a number of overlapping symptoms. So it's interesting that SRGAP2, in that sense, is a common link at the molecular level, at least, between SYNGAP1 and CTNND2. So that goes in line with this idea that I think many, many people are after, and which is, could there be some common pathway of diseases that looks different, and that could be then, of course, much more efficient to target than, you know, each individual genes, which is the way that, you know, we are all trying to do at this point. So CTNND2 SYNGAP1 is the first example of two proteins that are regulated in the same pathway that is linked to SRGAP2A and SRGAP2C. More work to do. Yeah.
More work to do. Thank you. Thank you so much for your time. You've been very generous with it. I know it's the evening there for you.
That's totally fine. I was very happy to be invited again, and I'm always amazed by the feedback that I get from you. So always a pleasure, and thank you again for your attention and all your feedback. It's a lot of support for us.
Thank you. I look forward to talking to you again in the future.
All right. Thank you. Bye-bye.
Bye.
Bye.